Hello everyone. The session today is everything about herbarium. I'll be including some of the historical parts and the essential components while preparing a herbarium and what are the herbarium units that is currently being you available in the world. So all these parts I'll be including here. So let's start. Firstly, we need to understand what is a herbarium. So they are literally a collection of the preserved plant specimens which are stored in a very controlled environment. So this is just a definition of herbarium. But before we understand how it is being preserved, how it is being stored, so we need to understand the history of how it began. So in the ancient periods, uh, like between the 3000 BCE or before the Common Era till 500 CE. So at this period of time, at this ancient period, there were different Egyptian herbalism, there were different Greek and Roman people, there were different Chinese people. What they do is they had a collection of medicinal plants and they wanted to document and they wanted to keep records of the medicinal plants, especially medicinal plants. So they have a description of plants, medicinal plants, and then they documented it in a different ways. Sometimes they kept the plants in a dried form. So but not exactly in the way the present herbarium specimens are being made. So this was in the ancient period between 300 BCE till 500 CE. So after that, there comes a medieval uh, period between 500 to 1700 CE. So in this period of time, there were monastic gardens, there were herbalism and apothecaries. So these were the period. Again, they collected plants with respect to spices. They for culinary purposes, medicinal plants with medicinal properties. So they preserved the plants. Like I said, they were mostly for the medicinal uses and spices. So after that, during the 16th century, that was the initial part where the herbarium are being, initiated. This was by Luca Ghini. He was an Italian scientist or a botany professor from Italy. So what he did was he preserved the plants in a particular way. He dried the plants, keep it under pressure, and then uh, bind these specimens within a book. So this is how he started preserving the plant parts. So for a long time, this has been going on at that period of time. So this was the first person, and this is how he preserved the plants. So he would uh, attach the dried specimen on one side of the book, and the other side, or in the same part, he would write the description of the plant parts for easy identification. So this is how he preserved the plant specimen. This had gone for a very long time. This the way he preserved, and there comes the century, that is the 18th century. This was the time when Carl Linnaeus he collected the plants in that particular manner for a long time, and what happens was he extended his collections, he expanded, and when he expanded such a system, it was difficult, to keep that bound specimen in a particular book. So that was a challenge for him. So later he started with an idea of mounting a plant specimen in a particular large sheet of paper. So this is the era of evolving the plant specimen in a herbarium form. So that is how he started the herbarium in a mounting sheet. So this is the history, and gradually till now you can see there are development in the herbarium facilities, in its storage, in a different way. And nowadays you can see that in many places you will find e-herbarium and all. So that was a part of the history. So now we will start with the basics of preparing a herbarium specimen. What are the components and what are the necessary things? Okay, I'll start with the collection, but collection is not the first part, because for collecting a plant, you have to you have to plan a trip. You have to go along carrying all the essential instruments or components. For example, you have to carry your camera to take photographs of the plant parts. You have to carry cicature. You have to carry uh, Caesar or something like that. You'll be carrying notebooks, you'll be carrying field data books. You'll be planning a trip is not an easy task, and especially when you plan for a long period of time, then you have to arrange a place, a stay, everything. So I'm not including those activities, but I will start with the collection part. So when you collect a plant for the preparation of herbarium, you need to make sure that the plant, it has to be healthy. So uh, there are different plant with a different habitat, right? So some plants might be aquatic, some plants might be a herb or shrub, or some plant might have a thick leaf character, fleshy leaf character, or a thick uh, fleshy fruit. At that period of time you have to make sure what is the best size to cut. So it has to be healthy, it has to have either leaves or fruit, flower or fruits along with the leaves. So all those should be considered, and it has you have to take the plant part with less dirt. So that is the initial part of the collection. And then uh, always there should be duplicates. So duplicates and more than duplicates, it should be collected, but it would be given with a different name in the later part of the class. So here in this section, the main thing is you have to take the healthy twig, healthy part of the plant with different plant parts, and that is the key here. So immediately after collection, it should be pressed using a blotting paper or uh, newspaper. But many times it happens that when you are in a long trip, tour, that is not possible, because it is difficult to carry the herbarium press. It is difficult to carry a lot of blotting paper with you. So at that point of time, in earlier time, what people did was the plant parts will be kept inside a vasculum. So in the vasculum is you know cylindrical uh, type of container where you would put inside the plant, and then close it, and you will carry till it is being pressed. But there are many challenges while carrying, because of the weight you'll be carrying around. So here and later there comes a point where people can easily store the plants in a polythene bag. So at that period of time, people slowly make use of the polythene bag. Big polythene bag, if you keep inside the plant in a fresh condition, you will the plant will last long. There will be no loss of moisture, and it will be in a healthy state till it is being pressed. So you have to make sure that it should be pressed as soon as possible. or right after the tour. So this is the important part of the collection part. So like I said, for immediate pressing, you need to have blotting papers, or you can make use of newspapers. Newspapers are easily available, so that is the easy pressing way you can do using herbarium press. Okay, so while collecting, it's not that we have to just collect the plant part. You have to carry a field data book. So in the field data book, there should be different information in the field data book, which include uh, the date, the location, and then the if there is any vernacular name, local name of that plant from the local people, if you can get it. You have to mention the habitat, you have to mention the vegetation, and the description of the plant. You have to write down the plant characteristic, any key character if you can find. So all these things you have to collect along with the collector's name. So it is important to fill up the fill data book, because many a times it happens that after drying the plants, if you do not write down all these details, uh, especially your plant characters, uh, it might lose after the plant gets dried. So it is important to write all this down, and taking a GPS is mandatory, uh, and then the date, the collector's names, all these are mandatory, and you have to fill this book. So for uh, for collecting the GPS, which is the global positioning system, so this includes the latitude. You'll be including the latitude, longitude, and elevation. You can get it from the GPS. So after that, the the stage after that of uh, immediate pressing is the drying and pressing of the plant until it is in the form of a healthy dried plant specimen. For that purpose, you can make use of blotting paper or newspaper, whichever is available, but the best is blotting paper. uh, and depending upon the moisture content of the plant, you have to make sure that it is being continuously changed. The blotting paper should be changed continuously. For example, when if it is a aquatic plant, you have to make sure that the plant is changed. The blotting paper or the newspaper is changed twice in a day. Or if it's just a normal herb or shrub with less moisture content, then you can change it in every day. So once it is changed, you should be pressed all together using a herbarium press. And herbarium press, you can customize it according to your requirement. So there are different types of herbarium press you can see from all different parts of the world. So one thing, during the pressing, you have to make sure that the plant parts are properly cleaned and all the dirt or muds are removed, because this might cause infection in future. And all these plant parts should be cautiously, displayed. If it's a leaf, then the both sides of the leaf should be displayed, and the flowers are arranged in a particular way. So the plant size should not exceed the size of the herbarium, which is 28 into 48 centimeter of dimension. So you have to make sure that the plant is in fits the dimension of the herbarium sheet or the mounting paper. uh, Sometimes if the plants, for example in the poaceae family, you'll find lots of long, plants. At that point of time, what you can do is you can put in such a way that it fits in a V or W shape. so that all the plant characteristics can be easily visible. So the next is, like I said, they have the blotters or the newspapers. They have to be changed every day to make sure that the excess moisture is being removed. So this continues for about 6 to 10 days, and often in a normal condition, the plant gets easily dried between 6 to 10 days, and it is ready to be mounted. But when there is a in rainy season or in a very humid condition, this is not possible, or especially if it's aquatic plants, it might get a little problem. So in those weather conditions, humid conditions, you can make use of the oven drying chambers. So drying chambers will help to remove excess moisture. But I can tell you that uh, without oven, without drying chambers, you will find the best herbarium specimen, because using the drying chambers, you'll find that many a times the color get this there is a discoloration of the plants. So that way, if you can find a way without using the drying chambers or the oven, then that is the best. So the next part is poisoning. So the plant specimen, the dried plant specimen needs to be poisoned, because in the course of time you will find that the plant gets infected with small pest insects. So for that matter, you have to make sure that it is being poisoned. There are different ways of poisoning. You can poison with mercury chloride solution. You can use lauryl pentachlorophenate or the LPCP. Then there is formalin in different concentration or in different ratios. And then you can there is a way of fumigating the place with poisonous volatile liquids like methyl bromide, carbon disulfate, carbon tetrachloride. These are ways to fumigate the plant or the surrounding environment. But the most common you will find in many of the herbarium unit is the mercury chloride. You can make a solution of mercury chloride using ethanol, 1 part of mercury chloride and 8 to 9 part of ethanol. So that ways you can make a solution, use a tub, and then dip the plant part, plant specimen, dried plant specimen, for at least 1 minute or 2 minutes, and then immediately you have to remove and dry it out. Otherwise, if you keep for a longer period of time, what will happen is the plant color get this like there will be a discoloration again here. So that should be kept in mind. So the plant parts or poison specimen should be then dried out for a few hours before mounting into a mounting sheet. So this is the poisoning part. Okay, once the plant specimens are poisoned, dried, then it is ready for mounting. And for mounting, what you can do is you can make use of glue, and then uh, neatly you can uh, apply the glue using a brush, and then you can attach uh, the dried plant specimen into a herbarium sheet or the mounting paper. So the mounting paper, you will find it as a thick paper. So again, you have to make sure that there is enough space for the label to be attached in the right hand lower corner of the herbarium. So it is very important. I'll talk about the label here in the later part. So like I said, the plant specimen, if it is longer than the mounting sheet, then it has to be bent in such a way that the plant parts are visible. Usually they are bent in V and M or W structure, so that all the plant parts are visible, and they are neatly spread. So these are some of the examples I'm showing you here. Some of them are our uh, herbarium from APRF, and others are from the internet sources. These two are from internet sources. Okay, so here you will find that there are different plants, especially when you if the plant is a tree, or if the plant is a shrub with a woody structure, uh, the it might easily come off. So you have to make sure that there should be independent stitches to be made in this plant. So how the stitches are to be made: you can pierce a hole on either side of the stem, and then you can run a clean thread in between the two holes, and then make independent stitches. So this is how the stitching is done. And then if there is any fruits, dry fruits, then it can either be attached directly to the mounting sheet, or sometimes you will find that those fruits, small fruits, minute fruits, are being placed as a pocket. You can also paste a small pocket on the mounting sheet, and inside the pocket you can put the small seeds. You'll find in some places that the orchid seeds are very minute, they're very fine. So yeah, such kind of fine seeds can be attached inside the pocket, which is attached to the mounting sheet. Okay, so let's talk about the level that I mentioned earlier. So there is a level to be attached at the lower right hand side of the herbarium sheet or the mounting sheet. And what this level should include: this level should have the details of all the characteristics of the plant, including your family, your genus, the species, and it is mandatory to have your GPS location, and then the plant description in a very short way, and then any plant notes, the collector's name, or if there is any local or vernacular name, your name of the organization or institution, and the voucher number. So all these details have to be included inside the label. So once it is done, the plant identification should be done. But I can tell you that the plant identification is not just a single process. It is a continuous process, and even if the plant is being identified and it is placed, later the plant identity can be, change, or depending upon the acceptance of the key characters. So it is a continuous process. So there are scientific methods of identification that should be followed. So by finding out through the key characters, comparing the descriptions, illustrations of authenticated records of data, and match with the similar or related herbarium of the world. So this has to be done during the identification process, and you have to make sure that the level should not exceed more or less approximately 8 to 12 centimeter dimension. So again, okay, before that, the level I mentioned about, there should be a voucher number. There should be a voucher number. So that should be attached to the mounting sheet. But again, there is an accession number. So what is this accession number is? It is in a register. You will be recording all the herbarium in a particular order, in a particular arrangement, in the almirah, right? So these should be in a particular order, so these order are uh, mentioned as a number in the register, so that we call it as an accession number. So accession number is provided to herbarium specimen which is dried, pressed, poisoned, mounted, glued, stitched and labeled. So the list of accession number of different specimens is registered in a register with date and stamp of the institution or organization. So you have to have a knowledge of the difference between the accession number and the voucher number. So earlier you'll find that we have mentioned herbarium number instead of a voucher number. So it is voucher number is equal to the voucher herbarium number. Voucher number equal to the herbarium number. Yeah. Okay, like I said, the plant identification is not a single process. It's not like you have identified a certain plant cannot be the changes cannot be made in future. So if there is any confusion, is not satisfactory for a plant to have a certain plant name, if it does not fit any satisfactory match and identity at that point of time, and if it's already been identified as certain species, at that point of time, what the person can do is you can make all the necessary changes. You can uh, identify the new you can put a new identity, but you should make sure that there should be a determination slip. So what this determination slip does is it will tell you that certain this specimen, this herbarium specimen, has the identity of this species is being changed by the certain person, and because of this particular character, so there will be a particular number for that, and the name of the person who has changed its identity because of the certain key character, or as the re-identification is done. So this is how it is a continuous process. So the herbarium specimens are arranged according to a recognized and accepted system of classification. So you will find that they are neatly arranged, uh, and generally they are arranged in a Bentham and Hooker system of classification, which are generally accepted in many of the Indian herbaria for its convenience. So you will find it in the such arrangement. Okay, let me tell you some of the essential components of the herbarium unit. So when you enter a herbarium unit, you will find all these things. And if you do not have, or if an organization wants to develop a herbarium unit, then that organization or institution is should have all these essential components for developing a herbarium unit. Basically, it should have an AC room, which is required for the deposition of the herbarium. And then there should be a customized pigeonhole cupboard made preferably of steel is required. Like I said, the cupboard should not be of different material or wood, because that might be infected with termites in a later period of time. So you should make sure that they are made preferably of steel. It should be a pigeonhole so that it is clearly visible from the outside. And then there should be dryers or drying chambers. And then there should be customized wooden herbarium press. There should be naphthalene balls that should be placed inside the almari cupboard, a pigeonhole cupboard. And then there should be blotting papers, genus cover, species cover, mounting sheet or mounting board, and then slips for identification, logbook, and all these are necessary uh, for these are the basic things that has to be there in a herbarium unit. So the blotting paper I've already mentioned. For the initial pressing of any plant specimen, you make use of blotting papers or newspapers. And then the mounting paper or the herbarium sheet is the paper where you attach or you glue the dried and poisoned plant specimen. So that is a thick kind of paper. And this mounting paper, once it is properly labeled and then glued, that is now ready to be kept, then it will be kept inside the genus cover. So inside the genus cover you will find different sorry, not genus cover, it should be inside the species cover. So in this, under this species cover, you will find the different habitats or the same species from different places you will find. And this species cover, now it will go under the genus cover, and then this genus will go under the family cover. So that ways it will be arranged according to the family and according to the different system of classification. It will be kept inside or stored in the herbarium unit. So this is a small picture that I am showing you here. This is the herbarium unit of CSIR IMMT that we visited in Bhubaneswar. They have kept it like this. Okay, again, I'm showing you two different herbarium here, the right side and the left side. So the right side is the herbarium of APRF, and the left side is the herbarium of APRF that is submitted to a national herbarium unit in Kolkata. So they have, you can even make use of color codes in the herbarium sheet. So many herbaria they make use of the color codes like this. So basically there are different types of herbarium specimen, and the difference in this herbarium specimen is very minute that it is quite confusing. But once you understand the different types of herbarium specimen, you can easily make use of those information, those ideas, uh, uh, while while your research or work is you know it will enhance your research activities. So firstly, I'll talk about the holotype. So when we say a holotype, so it is an original specimen that is used by the botanist. So for example, if you discover any new plant species, then you collect the plant specimen, right? So the first collected plant specimen that is known as the holotype specimen, and it is a single physical specimen of the plant. So holotype is a single plant specimen, and it is used by the botanist to uh, describe the plant for its characters. So it is also to ensure the accuracy of the plant identification and naming. So holotype is just one uh, plant specimen that is collected by the botanist to describe the new plant species. Next is the isotype. So it is not necessary that while collecting a holotype, you will be collecting only one species. So you will be collecting duplicates of it. So again, holotype, while collecting the holotype, the duplicate that you collected, that is the isotype. So it is a specimen that is identical to the holotype and the original specimen, which is used to describe the plants, and it is often used as a backup or duplicate. So for example, if the holotype specimen is being damaged or it is being lost, in that case of can make use of the isotype for checking its identity, for checking its plant characteristic. So at that point of time you can use the isotype. So in a very general way you can say that it is a duplicates or while collecting the holotype. So it is often when we say isotype, it is often collected by the same person. They are typically collected from the same location as the holotype, and it is often collected on the same date as the holotype. And in case the holotype is lost, damaged, or destroyed, the isotype can serve as a reference. So you can see it is a very minute difference between the holotype and an isotype, but it is a difference, of course. Okay, a scene type. So when we say scene types, they are types. They are a set of specimen. When I say holotype, it is a single specimen. When I say isotype, it is a single specimen. When I say scene type, they are a set of specimens that are used to describe a new plant species, and they are the original material used by the botanists to describe the species. So it's not necessary that you will be collecting only two duplicates or single duplicates. So they will be collecting a set of specimens. So that is the scene types. And scene type, if I say scene types, they are a set of specimens rather than a single specimen, and they are the original material used by the botanist to describe the species, and they are used to describe the characteristic of the new plant species. Okay, lectotype. So when if a particular specimen is chosen from among the set of syntypes to represent the species and provide a clear understanding of its characteristic, so that is known as the lectotype. So the lectotype is the one that is chosen from among the syntypes. Okay, so it is a single and physical specimen of the plant. By selecting the single specimen as a lectotype, the researcher can clarify the identity of the species, and then the lectotype provides us a physical reference points of researchers to study and compare with other specimens. So you can now understand that the lectotype is the specimen that is chosen from among the syntypes. So this is again a very minute difference. Now let's talk about the paratype. So the paratype is an informally designated specimen type that is part of the original material used to describe a new species, but it is not a holotype, it is not a lectotype. So why is there a paratype? So paratype, they are not again holotype, it is not a lectotype, but it is an additional specimen that helps to support the description of the species. So what it will help is if there is any character differences, if there is any ecological differences, if there is any distributional differences, at that point of time it will be helpful. So they can support the description of the new species by providing additional information, additional examples to find a characteristic of the species. So that is the paratype. So here I am showing you some of the uh, plant specimens during this drying process in a herbarium press. Okay, all these this will be glued into a mounting sheet. So this is the final herbarium that is made. You can see here it is attached with the information. So while drying you have to make sure that there should be a tag uh, of the plant, of where it is being collected from, the GPS location, and then the date, especially when the plant is unknown to the person. So it is important. You can see here the seeds are being glued, as well as it is being stitched along using a thread. And then you can find here the independent stitches. So this is the way it should be uh, done while preparing a herbarium specimen. So you will find very well known herbaria of the world. So like the Kew Herbarium in London, UK, Chinese National Herbarium in Beijing, Central National Herbarium in Kolkata, the National Museum of Natural History Herbarium Paris, Linnean Society Herbarium London. You will find Royal Botanical Garden Edinburgh and Herbarium in UK. So these are some of the well-known herbaria of the world. So that's all as a basic knowledge for components of herbarium and how you can prepare a herbarium, or what are the components that are necessary to have in a herbarium unit. That's all about it. uh, Thank you so much.